So we have seen what you can do after 40 years of research and where we got, and the next talk is about the future. Although from the beginning of quantum mechanics, people like von Neumann or Wigner or David Bohm were always thinking of biology in the context of quantum mechanics, because biology emerged on Earth with proteins at the nanoscale, which is the interface between the quantum and the classical world. And it's always been centered of quantum physics again. But pioneers such as the speaker we have today, Alexandra Olaya-Castro, who I met when she was in Oxford and I was in Oxford many years ago, are pushing this forward for new sensors, for imaging, but also for fundamental questions about the origin of life on Earth, and I think about the future of quantum computers. As you know, the people designing quantum computers in a very, I would say, conservative way of using quantum mechanics are trying to control the bits. But controlling the bits, quantum bits, is very difficult and takes a lot of energy, and the computer always collapses. And some of us think that maybe you should let those bits collapse and use quantum mechanics, probably like biology uses quantum mechanics for computation. I don't know what she's going to be talking about, but I think we welcome her. And about bridging quantum science and biology.
Thank you very much, Sonia. Well, it is a pleasure to be here. Thank you for the invitation. And as Sonia mentioned, I can't believe that it was 20 years ago that I was in the building just besides here in the Clarendon lab, where I first came across the molecules that I essentially fell in love until now. And I think they will be the love of my life, scientific life, for a very long time, because they are complex. And they're counterintuitive, and they force us as scientists to go beyond the approximations we like, because they are beautiful. These systems are beautiful in a very different way, not just mathematically, but in what they do, how they manipulate energy, how they manipulate all the mechanisms that they have at hand to produce different functions. So when thinking about bridging quantum science and biology, there are different aspects that we can look at. So we can look at understanding the quantum science that one might have within these biomolecules that you find in living organisms. What is the biology of the quantum dynamics that these molecules might exhibit? But also how we can use all the quantum tools and quantum science and technology that has been developed in the last 50 years to actually measure and understand these systems in a different way. Now, because of the time that I have here, I will focus only on the quantum science that you might find in these systems, and maybe a little bit on the biology of that quantum domain, because that's the fundamental science that we still don't understand very well. As I was having a conversation with the speaker from the MPL before, and he mentioned everything that physicists develop, it's moved to biology because it will be used there. Here, the bit that I'm going to talk about is the conceptual framework and the experiments that one might use to understand quantum behavior of these very complicated systems. Again, this 20 years ago, one of the papers that I read in that office in the Clarendon lab was this small review, but as valid today as it was 20 years ago by Dowling and Milborn, when they were explaining the first quantum revolution and the second quantum revolution. And our understanding of quantum mechanics has given us a lot of the technologies that we have nowadays. But what quantum science is trying to do in the last 30 years or more is to actually manipulate that quantum mechanical phenomena that is counterintuitive to what we see in our everyday life. And just to give you an example, this is an infographics that I took from the NIST, where they explain what we are able to do, or better, what scientists that work in the lab in experiments are able to do today in controlling and generating quantum superpositions in molecular systems. So it is possible to address individual parts of a molecule, cool down one part, shine a laser in the other part, create a superposition between the degrees of freedoms within a molecule, and then characterize that quantum superposition. Of course, this happens in a lab at very low temperatures. But the superpositions that are created there, which for me, the best way to understand them is imagine you have interferences, not necessarily like the ones that you observe in water, but interferences of amplitudes of probabilities do not last forever. There are degrees of freedom that we cannot control, and the interaction between the degrees of freedom that we are controlling and those that we cannot will destroy those superpositions. Now, that process itself is a quantum process that has no classical analog. We'll talk about that in a moment, but it leads then to remove the superpositions, and therefore we find what we find in the classical world. Now, just to talk about the capabilities that we have in the area of quantum science and technology, nowadays, and this is a group here in the UK, in Durham University, it is possible to create superpositions long-lasting in the time scale of seconds, no milliseconds, no microseconds, in the time scale of seconds. So nearly the time that you have to ask a question here. And these are superpositions, again, of relatively complex systems. These are the rotational degrees of freedom, so they are able to create superposition of these degrees of freedom that are cooled down because they do not interact with the environment that will destroy them. So we have that control, but these are still complex, but not as complex as the biomolecules that you find in living organisms. These biomolecules, which I am illustrating here, a variety, are systems of the size of a few nanometers. They are supramolecular complexes, which means that they have, for instance, do I have a pointer here? Is this one? Yeah, so I'll zoom into these ones, but essentially they are proteins that are bounding. In this case, they are bounding chromophores. A few of them, more of them, some of them just absorb photons and create excitations and transfer energies. Some of them absorb the photons and actually generate a charge separation. Some of them is just one chromophore coupled to the protein, but even that system exploits a variety of vibrational degrees of freedoms to produce particular functions. In other systems, you actually have, rather than the electronic degrees of freedom, here is the spins of degree of freedom. Nature has a variety of biomolecules that have different functions, but the dynamics that is relevant for that will not last seconds. It will last a few picoseconds at most, or a few femtoseconds in the cases that are interested. So these systems do not behave like those molecules that we love to have in the lab. These systems behave in a way that we cannot necessarily control them, but nature does. And we said that then these systems operate in that quantum classical regime, because when they investigate them with ultra-fast spectroscopy, we're able to see that they are not necessarily destroying superpositions in the tiny scale, very, very, very fast, but in a tiny scale that is comparable to the time scale of the functioning, the specific functioning of these molecules. So in the last, I would say, 30 years, there have been advances in two directions that allows us to map in time and space what is happening within these biomolecules. On one level, at the ensemble level, so when you have many of these molecules, but also at the level of the single molecule. So two of those icons that have allowed us to see what is happening in this nanometer scale is the optical multidimensional spectroscopy, which essentially tell us in time correlations between excitation frequencies of a system and the emitted frequencies, and that tell us about electronic motions in the system. But we're also able to do not exactly the same, but something close to at the level of a single molecule. Now, I am not an experimentalist, and these experiments are beautiful. But at this point in the scientific development of this field, what we need to be able to do is what quantum science has done with those molecules in the lab. Understand the quantum behavior of these molecules at an individual level in an unambiguous way. So all the theoretical developments, which is the focus of my research, is towards understanding that behavior and proposing experiments, new experiments in that direction. So the main questions that I have been addressing is essentially in this area, and proposing experiments that explore what is that quantum behavior for these molecules, and then trying to understand how then we go and explore the biology of that quantum behavior. What I do then is theoretical research in which I bring tools from very different areas. So I bring tools from quantum science. So my background is I am a quantum physicist with a background in quantum optics. But then I need all the science that is derived in quantum chemistry and physical chemistry to be able to propose approaches to address this problem. So I would like to tell you now what is the understanding that we have, some common understanding and some still open questions. So I don't have a lot of history in my talk, but there is this sentence that has always stuck in my mind that was said by Albert Szent-Györgyi, and who said that life is nothing but an electron looking for a place to rest. And this is indeed a lot of the science that I will describe here, but I would say that there is another sentence that actually will summarize the results that I will present, and is that life requires a sophisticated handling of energy quanta. Do you know who said that? Just me. Just a quantum physicist. And I will show you that the particular systems that I have investigated the more, which are photosynthetic biomolecules, do handle every quanta of energy at a time in a very sophisticated way that no man-made system does it in the same way. So if we learn from those ones and we're able to do reverse engineering to handle energy like they do, we will have a potential source for many different technologies. Two of the biomolecules that I am interested, one is this light harvesting complex 2, which is found in photosynthetic purple bacteria, which is an anoxygenic bacteria. The relevance of that is that it doesn't use water, but it uses other things to create sulfate there. But the interesting part of this molecule, it has this blue chromophore, so it's this set of chromophores and this red band of chromophores. The light is absorbed by the blue. Collectively, by all these chromophores, is transferred to the ring of red chromophores, and from this molecule is transferred to another molecule, and so on until it reaches the particular molecule in the photosynthetic system that does the chemistry. This is another biomolecule, very different from the light harvesting 2 in purple bacteria. This is a phycobiliprotein that you find in cryptophyte algae. While LH2 absorbs near the infrared, this one absorbs in the visible. The chromophores that absorb the light are very different to the bacterial chlorophyll here. They don't have the symmetric structure here, yet these two molecules behave nearly in the same way. What is interesting is that despite the apparent differences, the physical mechanisms that they are exploiting to transfer energy efficiently are very similar. And I would like to tell you what they are, if I can move. So just to summarize those first steps in photosynthesis, each of these light harvesting antennae will absorb light, and they will absorb it collectively, which means that the chromophores do not absorb individually, but they interact to absorb the light collectively. And that interaction and that collective absorption is something that we can only explain in a quantum mechanical form, particularly exploiting those quantum superpositions. Once this electronic excitation is created, of course, we have an image and exciton, so I just put a cartoon of what I think an exciton might look like in an image. It is transferred between different biomolecules until it reaches the reaction center. The reaction center is where the chemistry of photosynthesis starts. This process at very low intensities of light happens with an efficiency that is larger than 90%, which means that almost every photon that is absorbed will generate a charge separation in this chemical unit. No man-made system does that, and we haven't been able to build one that does this. So that's part of the interest in these systems. Now, this absorption that I said is collective between these chromophores is exploited by nature to essentially, nature has engineered a variety of light harvesting antenna for all the different photosynthetic organisms that you find on Earth that are able then to exploit a wide range of frequencies of the light available on Earth. We see that we have organisms that are able to absorb near the infrared, but also in the visible and in very, very low wavelengths. And the main explanation for this ability has to do with those collective absorptions of those chromophores in these organisms. And to explain that, we can only do it by explaining superpositions of the electronic states of the different chromophores. So quantum mechanics plays a fundamental role in explaining the absorption of the spectrum by these photosynthetic systems. So we have no doubt in that respect. But what biology does with those excitons is a little bit more interesting than just that. It just happens that these biomolecules are not necessarily, are in different biological environments. For instance, in purple bacteria, you find them in the photosynthetic membrane. But when we do experiments with these molecules, those biomolecules are isolated from the photosynthetic membrane using some chemical processes. And then we understand them with the experiments that they do in solution. But nowadays, we have the possibility of understanding these biomolecules in the two different environments, isolating them from the photosynthetic membrane or studying them in environments that are similar to the photosynthetic membrane. This is interesting because when they do experiments with the photosynthetic, this particular LH2 on the membrane, and when it is outside the membrane, they see a slight difference in the absorption spectra. You might imagine that this difference in the absorption spectra is too small to have any significance in how this molecule functions. This is misleading. In fact, what the experiments show and what the theory shows, which is what I've done, is that this shift in the absorption spectra comes with a very significant shift in the structure of the biomolecule and in the interactions of those chromophores, and therefore in that delocalization of the electronic properties within a molecule. And what this does is that increases the transfer from the blue set, in this case, from these blue sets of chromophores to the red chromophores that are more strongly compact, increases by 30%. So a change in less than 1% in the frequency or in the wavelength of the absorption leads to a 30% transfer, much faster transfer within a single molecule. And what we have done is that if you change the lipid composition of the membrane, then that lipid composition will control that delocalization and that shift in the absorption, and then it will control this transfer time. So biology is very clever. It has found ways of adjusting just little motions in the electronic components, little adjustments to the electronic structure, to then exploit a quantum mechanical phenomena to induce faster or regulated energy transfer. So a lot of my work is trying to understand precisely that quantum advantage. So if it increases the delocalization by a certain factor, how much advantage does it give to the function of that molecule? Now, this has to do mostly with the electronic properties. But these biomolecules are dynamic units. So they not only have electronic excitations being photoexcited by the light, but as these electronic motions happen, the nuclei motions are not passive and also are not just thermal. What this means is that some of the nuclei motions here play a more important role than what we thought. Most of these processes happen at room temperature, where kBT is the scale that you might think dominates. So everything is random, any superposition there might die. But it just happens that in these systems, kBT is comparable to other relevant energy scales in the system. From the physics viewpoint, what this means is that you have different phenomena competing. So you have the phenomena of destroying superpositions being as important as the phenomena or the mechanism that will maintain those superpositions in these systems. So I'm going to illustrate what this means in the context of this. This is a cartoon in which you have a molecule that the molecule has been excited in the red chromophore. So somehow you have an experiment in which you put different lasers, and you guarantee that with those lasers, you localize your excitation in this red chromophore. So you localized. The absorption is such that if you look at the probability of where the excitation would be, it has a larger probability of being in the red. And therefore, my experiment will consist in checking what is the time that it will take for the excitation to reach the green chromophore from which I will extract the energy. The 3 different panels that I have here will illustrate different interplays between different mechanisms. In the first panel, I'm just going to have interactions, electronic interactions between the chromophores. In the second panel, I'm going to have interactions between the chromophores, but also with nuclei motions of the chromophores and of the protein. And in the third panel, I'm going to have a lot of those nuclei motion, thermal motions dominating. Let's see which one gives us the best transfer time from the red to the green. So in the first, you do this experiment many times, and after all those times, you check what is the transfer time. And the transfer time in this situation where I only have electronic interactions is around 4 picoseconds. Now, in the second case, this vibrating or this motion like that is illustrated that now you not only have those electronic interactions, but you have interactions to the vibrational background here. And some of those interactions are collective. In that case, you see that the transfer time is around 1 picosecond. And in the final panel, you have a lot of thermal motion. So you ramp up the temperature of these systems. Any kind of phase will be destroyed. It's so noisy that what happens in that case is as energy is being transferred around, it is localized in each of these chromophores, so it takes very long time to transfer from the red to the green. So as you can see, this extreme and this extreme are not so good for the transfer of energy in the system because in both cases, it will take a long time. The mechanisms are different, but the end result is inefficient. It just happens that these molecules seem to operate precisely in this regime. When you have that interplay between having those electronic interactions between the chromophores and some collective coupling to some vibrational motions, and some thermal noise enough to guarantee that energy is transferred from the green to the, from the red to the green, fast enough, utilizing those cooperative behaviors, but also utilizing thermal noise to make sure that energy is actually transferred and it doesn't stay back and forth in this system. So that is an understanding that we seem to share. And what we want to characterize is what is quantum mechanically with no analogous in a semi-classical description for these systems. And this is what I've been doing. So I've been doing for a long time now, trying to understand what these mechanisms, what it does, is the advantage that these mechanisms give. So I will not go through all the results here, but I want to tell you, we have been able to show that these mechanisms actually are such that if you were to describe them quantum mechanically, you will find some kind of probabilities that are negative, which is a strong signature that you have superpositions during the dynamics of these electronic excitations. We have been able to show that you can understand some of the behavior in this system in terms of synchronization of local vibrational motions. And that synchronization shares some characteristics with synchronization in other parts of biology, but it is also different in a quantum mechanical form. From the function perspective, we have been able to show that that cooperative behavior and collective behavior during the dynamics allows us to understand that function in terms of being very directional, but that biology is still very clever again in that it doesn't always do the same, and it depends very much on how you tune interactions between the chromophores. So there is a rich physics behind these biomolecules that goes well beyond what you can control in a lab, in the systems, in the idealistic systems that you have in a lab. That gives us basic understanding, but these complex systems requires from us to go well beyond those approximations and envision other ways of controlling, because the ways in which biology control is not necessarily by lowering the temperature, but by making those small adjustments to structure and then changing function accordingly. There is another field, but before I go into that, I just activate it with light. It generates a radical pair, which is essentially two electrons that are paired either in a triplet or in a singlet conformation, and there is an interchange between these two. And that is the interchange depends on how it interacts with the magnetic field, and in particular with that direction, and that gives the advantage for that magnetic sensitivity. There are different studies around that, but one very recent, which involves people here in Oxford from Peter Hoare group, it is actually confirming that that is a very strong hypothesis for some of these migrating species. These experiments are in vitro, but essentially in vitro they are showing that there is a rapid interconversion between the triplet and the singlet state, and that this interconversion depends very strongly of the direction of the magnetic field, and therefore this is probably a very likelihood that this is a good mechanism for that magnetic sensitivity. What we have done is that we said, okay, if that is the case, I'd like to understand what exactly is the mechanism behind that. I'm gonna, I keep pressing the wrong button, sorry. I'm gonna go back here. Now, I need to go back to this one. Now, those spins are not isolated. These are spins that are meant to be, for instance, here in the cryptochrome, so it's a big molecule. So they are embedded in an environment. They are not just simply isolated. So for me, the main question is whether that environment is helping that magnetic sensitivity. So we try to model the interaction between these spins and the environment in which it is embedded. We don't know all the details of that environment, so we just model it in a general form. And we want to understand whether correlations between the spins and the environment will give an advantage for detecting the direction. And what our studies have shown, and some other studies from other groups around the world, including here in the UK, are indeed showing that if you consider not just the dynamics of that pair, but how that pair relates to that biological environment. So in this case, it's the cryptochrome. There will be other spins there, but there will be other mechanisms within that protein, vibrations, et cetera, that will couple to those spins. And that coupling between the spins of interest and that environmental, biological environment is not simply destructive. There will be some collective behavior that lasts enough to ensure that it is used for detecting the direction. So this is a quantity that tells me about that direction, and this is a quantity that tells me about that collective behavior, not just of the spins, but that biological environment. And when in our studies for different characteristics of that environment that we don't know, but we just assume different possibilities for that environment, we always see that the sensitivity to detect the direction of the magnetic field depends very strongly of that coherence. So what this is telling us is that biomolecules have these particular mechanisms that go well beyond what we are often used in a lab with very cooled down systems to exploit what we thought it wasn't exploitable, to guarantee that quantum phenomena does affect function. What I would like to do now, and is the processes that I have teamed up different experimentalists, is I would like them to show that unambiguously at the level of a single molecule. But that will be for another time and another talk. So for now, I just would like to finish with some summary. Understanding the quantum domain of biophysical and chemical systems has led to already advances in biophysics by relating quantum dynamics and its non-classical features to function. We still have a lot to do there, but we are already doing science in this direction. It has also led to new ways of describing and observing biomolecular systems. It has led to a much deeper understanding of the quantum resources that we have in complex systems, and it is definitely linked to the advancement of spectroscopy to resolve electronic and nuclear in space. And none of this would have been possible without all the early career researchers that have trusted me and that I work with and that I enjoy so much, and the funding of the people that believe in the work that we do. Thank you very much.
questions? There. I have questions.
Thank you very much. I'm going to ask you an interesting question. If you're working from a quantum theory and it's a closed system, how would you open that relative to the biological system you're trying to open?
Sorry, can you...
So you're using a quantum system which I believe is closed, is that correct?
No, I use open quantum system description.
Okay, so how are you opening the quantum system and the
So
biological system in parallel?
Okay, so what we do in this biomolecule, the open quantum system description, what it does is I focus on the degrees of freedom that I know we can track in an experiment, like in spectroscopy. So in this case, because it's an optical spectroscopy, I will focus on the electronic degrees of freedom. But in the mathematical description that we use to that, any other degree of freedom, like in this case, the vibrational degrees of freedom, that are interacting with those electronic transitions are treated in a way that I average, but I keep track of the correlations. And then I focus not just on the average of my electronic observables, but in the fluctuations of those. And we know from quantum science that whenever you focus on fluctuations, that will give you a lot of information about those correlations. So everything that I do here focus a lot on two-point correlations and fluctuations to understand that influence. And we treat the interaction between that in two different forms. Sometimes we do it perturbatively, just to understand any possible insight. But for predictions, we treat it in a non-perturbative way, which means that I account for those correlations in different degrees as I go along in the dynamics. Thank you.
Here's a question.
Thank you very much. Absolutely brilliant talk. Some nice quantum mechanics underlying that. Question I've got is one about the universality of these sorts of mechanisms. Clearly they have evolved, so it is, in terms of competition, a good thing to do. And my question is, you've seen it in light gathering. Is this sort of mechanism where quantum effects fine-tune the efficiency of processes likely to be pervasive in most energy-related activities in cells and organisms?
This is the 1 million question, to tell you the truth. And I have to be completely humble here and say that the answer is, I don't know. I would like that to be the case, but I don't know. So what we need is experiments that do that. So we team up with two groups of biologists. One is at the moment at Imperial College, and he's an expert in laboratory evolution experiments. And the other group is a group at Surrey University who is interested in this problem from manipulating, doing site mutations in these molecules. The experiment that we will need to do for this is, well, I don't know the universality, but to understand whether evolution is tuning this and actually exploiting is to do a laboratory experiment in which in vivo, we see whether by doing some modifications of this, we have a competition within a species, and the species that wins, let me put in this language that is not the biology language, but how I summarize it, then extract the proteins from that, and then analyze if in that case we have some kind of increased quantum behavior in comparison to the one that didn't win. Those are the kind of experiments that we will need to do. And I tell you, we are in that direction. But the answer at the moment is, I don't know. I would love that to be the case. From the physics perspective, I can understand exactly why that is the mechanism that we'll exploit. Because when light, for instance, illuminates a particular system, it doesn't excite everything. It just excites certain degrees of freedom. But those degrees of freedom are always interacting with that environment, which essentially means that a dynamics will be inputted just by these non-equilibrium processes that happen. So it seems very natural to think that those correlations between the system that I'm interested in and the environment will last for some time. Whether it is enough to affect biology, I don't know if in all the systems, but it definitely be present in many biomolecular processes. Those that are activated by light and those that not. Thank you for the question.
And then one there at the back.
What do you think will happen if and when the magnetic fields flip over from north to south? To the birds, I mean, not to us. Is it going to be a disaster for all these?
I think it's just going to migrate to stay there for a while and then move. I really don't know. I'm sorry. You just remind me, I went to give a talk somewhere else, and I talk about the photosynthetic system. And there was this gentleman on the audience who had been observing swans for a long time. And he asked me a question related between the swans and the photosynthesis. I couldn't give him a proper connection, but I know that his thinking was absolutely in terms of trying to understand the molecular origins of whatever was the behavior of what this bird was doing. Yeah, no photosynthesis, sorry, no photosynthesis, the bird navigation here. But I couldn't give it because I don't know enough of the biology there. But I do think there are these principles across the biological organisms that we still don't understand all fully. So I cannot answer just because I don't understand it fully, but I'm sure there is an interesting question there. Thank you.
Hi. Thanks for that fascinating introduction. I'm at the back on the camera.
I can see you. I can see you. Yeah, sorry.
Yeah. Well, I'm no expert in quantum mechanics. I'm an engineer. I'm just trying to follow what you were saying about molecular vibration, improving the efficiency of light gathering. So are you saying there's a kind of a sweet spot vibrationally with these, are they called chromophores?
I think so. What the experiments are showing here, the ultra-fast spectroscopy experiments that are showing here, is that when energy is being transferred within the molecule and it is involving two different electronic states, there are often vibrational motions that will have a quanta that can commensurate with that. And that, therefore, if there is this resonance of energy scales, the system will exploit that to use that transfer pathway. And therefore, it will help to transfer faster, but not just faster, but in a directed way.
Right.
So there seems to be this resonance. But even when the vibrations are not necessarily on resonance, because you have collective behavior between the electronic motions, it will exploit a variety of vibrational motions just because of that delocalization.
That sounds very complex as an ensemble.
As an ensemble, definitely. Therefore, we definitely need to go and do single molecule experiments to try to understand it there. But the ensemble experiments are already showing that that happens.
Thank you.
A bit more of a personal question. You said at the beginning that you fell in love with quantum mechanics. What was it about quantum mechanics that made you fall in love with it?
I didn't hear. Sorry, I didn't.
When at the beginning you said that you fell in love with quantum mechanics.
Quantum mechanics, yeah.
Yeah. What was it that made you fall in love with quantum mechanics?
Do you understand that? What was it made you fall in love with? What makes me fall in love? Okay, sorry. I was like, I think this is something very personal, and it's the fact that when I was doing my PhD, I remember being at 3 in the morning in that office in the Clarendon lab and having a figure in which I was predicting the correlations between some cold atoms in a cavity, and the prediction was exactly what I expected. And I felt very happy, but I also felt disappointed because I felt, this wasn't that difficult. So I felt like I needed to answer questions that were beyond my, not that they would be very difficult, but just I needed a question that was more challenging. So when in that time, the supervisor came and told me, look, do something about in this field, then I said, I'm going to do something about this in this field. And I'm 20 years after, I'm still trying to do something. I haven't done much, but I keep trying to do. So what made me fall in love is that they were very counterintuitive. And for me, this counterintuition means that I'm learning at all times about those systems. And to the rhythm of what I learn, my heart palpitates. So that's the reason.
I can add something to that. There are many women working in quantum biology, actually.
Yeah.
And I think maybe because we're in minority and we went to physics against the tide, against the odds, we were always misplaced. We like complex systems. I am a biological physicist, and we don't like the experiment or the computation, like you say, that fits the model, maybe because we don't fit the model and we know there are other ways of living, and we just want to look for the things that don't fit the model. Yeah. I see that very often.
Yeah. It can also be, but adding to all of that, it's just the fact that maybe this is more a philosophical, but I remember thinking, we are explorers, so we go outside the Earth and we want to explore what happens outside the Earth, but we don't even understand fully what is happening on the Earth. So that for me, I am an explorer and I support fully that explanation beyond Earth, but I do still want to understand what happens in this beautiful Earth. So that's as well.
More questions. We have a bit more time. I can make a question if nobody has a question. I have a question.
Yeah.
So all you guys, all of you, but many of you working in quantum biology, you're very worried and interested in correlations and matching that. It's mainly quantum chemistry and trying to see if you can then reach the biophysics. But biophysicists like me were actually very interested in quantum collapse. And actually we're interested in how biological systems might be able to extract energy from quantum collapse. For example, many of us are actually looking at if biological systems actually correlate in order to create clever noise that can be used for information processing in the context of computational biology, and for example, in the brain is this unaccounted energy of the brain. The brain only uses 20 watts. And we don't know how it does it. And some people, and many of us, think that it might be using quantum process, not only to lower the information, the energy that you need for information processing, but also being able to extract energy from quantum collapse. I wonder if you could comment on this.
It sounds like the open question for a research grant. But yeah,
yet another time.
But what I think the main comment that I would make around this is that what we are definitely starting to understand better and better is that how biology uses noise at this molecular level is a clever way. It will use noise to tune things. It will use noise to get out of that tune if that function that
emerges from noise in all biological systems. Yeah.
So I would say, and from the quantum mechanical perspective, we know that what noise does is that it will collapse those superpositions. So from that perspective, but from there to extracting energy, I'll have to make the full logical connection.
The quantum collapse models predict that just the collapse produces energy, and that's what we're interested in. There was an extra question there. Maybe there's a quantum collapse expert there at the back.
Once you've understood this process in a few years' time or many years' time, do you think maybe you could synthesize it yourself as a means of storing energy? Because storing energy is almost impossible, or it's difficult and inefficient.
Definitely one of the drivers to understand this is essentially manipulation of absorption, transfer, and storage of energy. The process of storage energy within the photosynthetic context happens in the reaction center, where you have actually not just absorption of energy, but you generate a charge separation, and from there you extract that, let's say in oxygen, the photosynthesis, the electron. So, and in all those steps, there are quantum mechanical phenomena involved. In the reaction center, similar correlations of the ones that I've described here are hypothesized as being present there and helping that storage process by directing energy to the chromophores where that will happen. So the summary is I think we are building all those we're having insight into those building blocks that will allow us to then do that reverse engineering and help that problem that you are asking. Okay, can we engineer this system with a little bit of correlation that will help storage better than if I didn't have it? So I think we're building, and engineers are working in that direction, and synthetic biologists by modifying, making kind of chimera, light harvesting with other things that will help either the absorption or eventually the storage. I don't know if that answers your question, but thank you.
We have time for one more question, or everyone wants to go to the tea break that I think we have now. Tea
Thank you very much.
Thank you so much. Question somewhere. Who was the question? There. It's so invisible under the light. But yes, but you're going to have to shout because the microphone.
So I get the feeling that quantum biology is an area that transcends two completely different fields, quantum physics and biology. We have biologists who are written about quantum physics and vice versa. How do you think, if you agree, that this challenge can be overcome?
Here, I can see very well, but I could hear your voice very well. Is higher? Oh, sorry, because you are under the light and I couldn't see you. Yeah, so actually this bridging between quantum science and technologies and biosciences is more just than quantum science and biology. It has engineering, it has physical chemistry, it has quantum chemistry, and it's part of the excitement of being in this field because it's essentially those problems in which we understand that to be able to solve it, you do really need to have the input from these different scientific disciplines. The main challenge there, you can imagine what that is. I'll give you an example. When I talk about coherence, the 4 or 5 scientific communities that are involved will not necessarily understand quantum coherence in the same way and will not have the same insight into how you go and measure it. So at the moment, the most basic, specific challenge that we have is that for the next generation of scientists addressing this, they need an education which has taught them from all these different perspectives how to think. I think in the previous century, we specialized so much. You know, we have the physicists, the chemists, each niche protecting their own knowledge. I am more intelligent than you, all of that. The problem at the moment is that we need to get out of that. We still need that specificity, but we need to understand how to talk to all the other communities. It is happening. But for me, that's where the challenge is, in bringing that conceptually. Once we understand each other, I am sure that the progress will be a lot faster.
Or to bring biology to a physics department, which
Oh,
is
exactly.
where some of us do.
Keep bringing them.
And that makes it easier.
Yeah. Right. Thank you. Thank you very much.
Thank you so much.